Hello, everyone. Uza and I are here today for another episode of Sambai Speaks. We are so excited to bring this series to you, this webinar series about the project that we are working on with our good friend, Dr. Melissa Davis at Morehouse School of Medicine, called Sambai. It's a Cancer Grand Challenge, and we are working diligently to talk about the disparities for cancer globally, for breast cancer, for prostate cancer, and for pancreatic cancer. So if you haven't seen our earlier episodes, you should watch them. But in the meantime, today we're going to be talking about our third work package about genomics. And Laura, our precious blessed Laura, has been serving on this team as our patient advocate, where we have patient advocates on every Sambai team. And Laura's here with us today because she kind of brought the patient voice to this really important work. So here we go. There's a lot of science here to learn, but it's all good stuff. And we're going to break it down to you and give you a good understanding of the work that we're doing because it's so important. It's going to change all of our lives. So Laura, so glad you're here with us.
Thanks for having me. Thanks for letting me be part of this really important work, groundbreaking work, really not being done anywhere. So it's a pleasure to be here. I'm Laura Crandon. I'm a blessed, so breast cancer that spread to my brain in 2018. And I started Touch for Life to share the experience that I had and the access to really important and advanced medicine that I had. And I thought it wasn't fair that just I had this access and these testing of genomic testing. We're going to talk about that, genetic testing. I realized that it made me a unicorn amongst Black people, and it really shouldn't. So started Touch for Life and Black Genes Matter to talk to our community about the importance of these two things to make sure that they have access to the most advanced treatment.
And who are you here with? Who's on your team, or whose team are you on with our scientists? So we have a patient.
Yes, I have the pleasure of being on Work Package 3 with really smart people. I don't know how I got on this team because these people were super smart. So Dr. Nicholas and Dr. Nyasha, I have the privilege of being on the team with them, and they're going to introduce themselves and the work that they do in leading this important team. And I get to bring the patient voice, the voice of you, the people who are watching this Sambai Speaks, to make sure that the everyday person's voice is represented on this team. So with that, I'll first turn it over to Dr. Nyasha.
Thank you, Laura. And thank you, Ricky, and everyone on your team for inviting us to Sambai Speaks. I've been listening, and so we're excited to bring a perspective from our team, the Genomics Work Package 3 team, here today. So my name is Nyasha Chambwe. I'm an assistant professor of molecular medicine at the Feinstein Institute for Medical Research. So this is a research institute that's embedded within a hospital system, which is Northwell Health in the greater New York area. So we're primarily in Long Island, Westchester, and growing. So I have a small research group that started about 3 years ago. And my training is in computational biology. That's what I got my PhD in. And so we use this discipline of computational biology to analyze the data that comes from genomic testing. So genomic testing generates large volumes, terabytes and terabytes levels of data. And so we have data scientists that work in biology, and that's what we do. And we analyze these genomic data to try to understand what's going on in patients' genomes and how that's related to their cancer and potentially what treatments they should get. And so that's the type of work we're bringing to Work Package 3. And we're going to talk more about that today, I think.
Yeah, we are, because you use some big words. So, but we'll go through them. We'll let Nicholas introduce themselves and we'll go back to some of your big words and try to break it down to words we can spell, but we're going to get there. I know you guys can explain it. So, Dr. Nicholas.
Hi, everybody. Thanks a lot for inviting me and for everything you're doing in the Sambai project. So my name is Robine. I'm Director of Computational Biology at the New York Genome Center. So I don't work at an hospital, but I work at a place that is specialized in generating the genomic data. So we have instruments, we receive samples, and we transform those samples into data that we share with our collaborators in all of the major research institution and hospitals in New York, but also outside of New York. So we were founded 12 years ago, or now 13 years ago. And I joined as one of the first employee at that time to work in the team of bioinformatics or computational biology. I now lead a team of 13 people working on cancer genomics and functional genomics, and we can discuss those differences later on. Um, my work applies to all cancer type. And so we have done work in all of the major cancer type, from rare cancer to pediatric cancer, more common cancers in all sort of different contexts and experimental design. But I'm really, really glad to participate now in the Sambai project. After spending a few years working on a project we can discuss also called Polyethnic 1000, where we study the cancer profile of patients of non-European ancestry recruited in the region of New York. And we have been doing that for the past 4 or 5 years. And that led to, or that was one of the preliminary data that led us to obtain this cancer grand challenge called Sambai, that is specialized on 3 different cancer types: breast cancer, prostate cancer, and pancreatic cancer, but really focus on patients of African ancestry. And so our work is really to transform the large amount of data that come out of the sequencing machine into smaller files that we can share with our collaborators, with the biologists that we work with the doctor we work with. And so compressing that data into information and to results that ultimately can be useful for the patients. That's a lot of what we do every day.
Very great. Okay, so I'm gonna take you back for a second. So we all kind of know what genetics is, and if we don't, so basically help me with this. Genetics is the things you get from your mom and dad that define your hair color, whether your hair is curly or not, your eye color, like all the things about your body that were passed down from your mom and dad. Is that the overly simple definition, right? So anyone want to add on to that? But so then let's talk about what genomics is. So when we're also more cancer patients, for breast cancer, they will often give us a genetic test. And I don't even want to say often, because a lot of Black women don't even get a genetic test. We know that, Laura, right? So we have to ask for it, but you should get a genetic test if you're diagnosed with breast cancer. Really any cancer at this point. But anyway, so we kind of understand what that is, but what is genomics? What does that mean? What does the word genomics mean?
Nyasha, do you want to start or you want me? So the way I would describe is the genome is the molecular information. It's the molecule, really the physical molecule that is present in all of our cells, and that our 2 copies, one coming from our dad, one coming from our mother. Okay, and we have that in all our cells. What we say by genetic test is usually testing a few small regions in the genome that correspond to a gene, okay? A few genes, okay? So the genome is 3 billion letters splitted into 23 different chromosomes, okay?
What's a genome? What's a genome?
So the genome is this set of 3 billion letters, nucleotide, but we can call letters A, C, T, and Gs, okay?
Okay.
And there are 3 billions of them, right? It's a large number. And there are a small number of those that code for what we call genes. So it's a smaller set of nucleotide, of instructions, of letters, and there are about 20,000 genes encoded in the genome. Okay, and they
Okay.
code for all of the function of the cells, for all of the protein that work in the cell and make each of the cells different. Right. So from the same set of information, we have different cells in the skin, different cells in the lung, in the liver, in the breast. All of our tissues are made of different cells that are different set of instructions that have different activities, but all of it is encoded from the same 3 billion letters in our genome.
They all have a different job or role.
They have a different job, right? They play a different part of the music if you want.
Okay.
And when we say a genetic test, we only look at a small number of those instructions, those regions in the genome that we know how to interpret. So we know some of those proteins, some of those building blocks very well. And we know that when there is a small difference in the letters, they give an additional risk for a particular cancer, right? So we know that in the general population, the set of letters is very similar, right? But then some people have a different set of letters at that particular location, and that gives them an additional risk for breast cancer. So when we do a genetic test, we test those regions in the genome. Out of those 3 billion letters, we test a few, tens or hundreds or thousands, but a really small portion of them. We know how to interpret, we know how to calculate the risk associated with those differences in those particular region, and that's what we call the genetic test. But that's very different from the genomics, which would be reading those entire 3 billion letters from one end to the next, to the end. Okay? So we take everything, and then inside those 3 billion letters, we can look at those regions we know very well, and we can get the same result. But we also have all of the other set of information that for now we are struggling to interpret. We don't know most of those places. We don't really know what they mean for the majority of them. But at least we have the vast majority of them we can identify through our sequencing platform, sequencing instrument.
What's a sequencing instrument?
Right. So, Nyasha, you leave me doing the New York
I'm
Genome
nodding
Center.
because
In
in this particular instance, this is what the Genome
This is
Center does every day.
Yeah, absolutely. So, as I said, we have the genome is present in all of our cells. And so we have machine that take those DNA molecule and read them. Really think of them as reading them. Okay. You
Reading
take
the letters.
one
reading
of
the-
them and you read the letters. There are some technical differences, and we talk about coverage, we talk about error rates. Let's not get into those for now, at least, unless you want. But it's really just reading. Sequencing is reading the genome.
Okay.
Okay? And we can sequence a genetic, for a genetic test, it's also sequencing, but it's sequencing only those regions that we know. We read them. There is an A, and then there is a C, and then there is a T, and then an A and a C, and so on and so forth. And we do
And you
that.
look at the sequence of the things.
So
All right.
it's linear, right? It's a molecule that one after the other, and they are attached chemically. Okay, and so there is an order in those molecules, and so we walk through them and we read them one at a time. That's what we
Okay.
do.
So when I got my genetic test, I spit in a cup.
Yep.
So that stuff that you're looking at is from my spit.
Yep.
Got it. All right.
And so that your spit contains some of the cells from your mouth. You
Right.
would have the same information from the blood. You would have the
Okay.
same information, by and large, you would have the same information from every cell in your body,
So like a piece of my hair.
piece of your hair.
What about a piece of your tumor that
Right.
you
That's
know
that's
is
that's the right question. And so
Okay.
that gets into the difference between what we call the germline genome, that's inherited, that's
Did you get
from
from
our
mom and
parents,
dad? Okay.
that we have in all our cells,
Okay.
and the tumor cells that accumulate mutation, right? And they have a certain set of mutations, so differences between those cells and the other cells in the body, and they accumulate mutations over time, right? And so that's the difference. But they also contain all of the normal genome, but in addition to that, they have particular mutation, right?
Bad
So
stuff. The
those
bad
mutations
cancer
are
stuff.
coming from, for instance, from the environment, right? We know that in lung cancer, smokers have a particular set of, or have a particular pattern of mutation because the tobacco is a mutagen and creating mutations, right? And so that's one cause of the mutations in the lung cancer, right? UV, for instance, would be another pattern of mutations. We know that melanoma has particular pattern of mutations coming from UV exposure, and so on and so forth. And so now we can, identify the genome in the tumors and try to learn something about what that tumor has been exposed to, what that patient has been exposed to, that will explain some of the behavior of the cancer cells.
Got it. Okay.
So I really like that question, Laura, because this is really that gets to the heart of what we do, and how genomics makes a difference in terms of how your cancer is classified, whether we know whether it's aggressive or not, and potentially what treatment you might be put on. So as we've been talking now, we've talked about how our cells, we have all the same information across all the cells in terms of the genetics, the genome, right? In the normal cells. But with cancer, what happens is that some of the normal processes in cells go wrong. So there are two main things. We have what we call a brake system and an accelerator system. So we have systems in our cells that stop cells from dividing uncontrollably. So that mechanism is broken in cancer. So it's called a tumor suppressor. So instead of saying, oh, we've divided enough, we form new hair cells, everything is fine, it's time to stop dividing, the process goes wrong. And now you have uncontrolled cell division. And that's what results in the lump or the growth that becomes a tumor. And
Okay.
that's part of the somatic genome, which is something has gone wrong. So we've acquired new mutations and we've formed something that's other than us, but it is based on us. So the pattern of those tumors is somewhat similar to your original genes, but has a few things that have gone wrong and have co-opted the regular program. Okay?
What causes that?
Hmm?
What causes that? But that's what we're trying to find out.
So that's some of the unanswered questions in cancer. So some of it is just plain bad luck. Okay, some of it is that there's this theory that mutations happen at random all the time, and they can happen anywhere. But if your mutation hits right in the brake mechanism, that's when you have this uncontrolled cell growth. There's also evidence that the mutations that cause cancer keep recurring in the exact same place. So we're like, ooh, maybe it's not an accident or by pure chance. They have these places that we call hotspots. We keep finding them mutated in cancer cells, in the cancer genome. And those, we can relate them to how aggressive the tumor grows. So you'll hear certain numbers technical terms being said that, oh, they have this type of cancer with this mutation, and it's a well-known mutation, and that mutation might mean that cancer grows super fast, or that cancer responds really well to a particular drug. And so that's kind of what we're trying to do when we're studying these genomes, and some of the work we're planning to do in Sambai is to understand what all the mutations are in these tumors and see which ones keep recurring and try to associate them with what happens with the cancers and in the patients.
And maybe see if similar hotspots exist in certain types of people, right?
Right.
Yeah, that's what I was going to ask. So I could have a mutation in my cell that could be the same as Uza's and Laura's.
It could
Yes.
happen in the same place in the genome, right? Like if we think of the genome as a book, we're reading it, right? That we're reading that sequence. It would be page 63, paragraph 2, word 3, position 6. So it'll be in the exact same place.
Okay.
But most of the other mutations will not be the same. Right?
Okay.
But there could be some that are the same, and they may be if on a large number of patients we have the same mutations occurring multiple times, that tell us something about the function of that mutation, right? If it happens randomly anywhere in the genome, then that's a different type of information. That's more what we would call a passenger mutations. It may not provide additional benefit to the cells. If they happen recurrently in the same place, then that probably gives an advantage to those cells and they proliferate more. And the cancer have some similarities because they are mutated in that particular genes, right?
Okay.
So only these genes would be the same, but the other ones would be different. But that's where if we find a targeted therapy that would hit this particular mutation, that would benefit all the patients that have this mutation in their tumor cells.
Okay. What's happening is that spell check that normally would say, like on Word or Google, whatever, that says, hey, that word is spelled wrong. instead of the ACT or whatever, it's ATC. It's just not catching them all, and it's how badly the spell check is misfiring. Like, is it on every page, every other page, every 20th page, or whatever? It's saying, hey guys, get in here. Like, we're gonna misspell everywhere, you know? And it's
So
how
that's usually,
well
yeah.
that's functioning or not is saying how aggressive that, you know, cancer or tumor is able to proliferate or, you know, grow and replicate throughout your book analogy. Or sometimes I use a gate analogy, like how wide is the gate to let those minions in? You know, just kind of come on in because the gate's supposed to be closed, but it's open, and it's how quickly it lets those bad guys in, type of thing.
Okay, so
That was a great visual, Laura.
yeah, that was great. So we have this genomics, and so are there things that you think are happening that you're testing for, or are you just trying to find it out? Do you know what I mean? Like, do you have like hypotheses or things in your head? Well, we think this is happening, or the disparity is happening from Laura cell to my cell or white to a black or whatever because of these 4 things that we're going to test for? Or are you looking for those things right now, or both? Do you know what I mean?
So we're doing a little bit of both. So in the clinic, they are testing for the well-known ones that we have reams and reams of evidence, because it makes what you decide to do implicates a life, right? And so
And
they,
the clinic means where patients are.
right, where the patients are.
Okay.
But in the case of Sambai, where we're doing a research project, we are trying to find new things that could potentially work better in the clinic. And so we are testing, we're measuring the genomes for, we're measuring everything and looking specifically. So we will generate data in Sambai for all our study participants, for the whole entire genome of each patient. And we'll store that data and reuse it multiple times to test different hypotheses.
Their
So
blood,
idea,
the patient's blood.
the patient's blood. So if we have an idea that let's look here, we'll use the same data because we'll have measured the entire genome, and then we can always go back and look and mine that data over and over again to test different ideas, or to look at different places in the genome that we might have an idea might be involved in a particular aspect of their cancer.
Nicholas, you were going to say something?
No, I mean that's exactly right. So we both can, test hypotheses, but also take an hypothesis-free approach and
I just
discover
love-
new things and new reasons why, that would explain some of the disparities that we are observing. I think one key advantage of Sambai compared to many other study is the interplay between the different components of Sambai. So compare, having the genome information for all the participants, but also all the questionnaire that will be collected in Work Package 1 and in the category of what we call social determinant of health, exposomics, what are
Right.
the small molecules floating in the blood that can inform on what the patients have been exposed to,
Right.
and for a subset of the cohort, an immune profiling of their tumors. And so relating all of those bits of information will be really, really critical and is very, very unique to Sambai and really has not been done at that scale anywhere in the world.
Wow. Wow. Laura, you had your hand up. Don't have to put your hand up.
The second part of this is the most exciting for me because heretofore to date, all the data, all the research has been based on non-Black people. So the reference point is always starting with people from Sweden.
Right. Sweden.
I'm like
Or
laughing, but like
Utah, right? Or like
yeah, like Utah, that's 800 women from Utah. Like that's literally true. And so sometimes when you get these, even if you're fortunate enough to get the results, get tested and get the results back, a lot of times as a Black patient, you get this result that's called VUS, this variance of unknown significance. So that it says there's something there, but we don't know what it is.
Because
I call it the very unsatisfying result, VUS, right? But the reason they don't know what it is because the reference data set isn't based on us, it's based on Swedish women. And so with this data set, what Dr. Nicola and Dr. Nyasha is saying is that the reference point now becomes us. And so
Black people, Black people.
yeah, Black people. And so the variance will potentially become a known area of significance that we should build a hypothesis around that says, gosh, this is an area that really needs to be targeted for people in the diaspora, Black people, people
But
with
then
African
or it's different for Black people versus white people, that we're seeing a
Yeah.
difference, right? And I think that's what you said, Nick, earlier too.
Sorry,
Go ahead, go ahead.
really touched, Laura, by what you said at the top of the of a session about the fact that you were tested in your journey. And I think this issue that we're discussing is of particular importance to everyone involved in Sambai and has been our calling card to why we've come to this project for a lot of us, because it's been very deeply dissatisfying to work in the field where we have data sets that are not as diverse as they can be. And to know that there are segments of the population globally that will get those, I'm going to steal that from now on, very unsatisfying sort of results.
Yeah.
And there's a historical component to this, so what we know to this point about which variants are associated with your cancer risk are stored in a database, right? So people have to be sequenced, and then we have to observe these mutations, sorry, not variants, mutations, and say, oh, these mutations seem to be associated with getting the disease or getting cancer. And so then that one is stored. Oh, we've seen one example of this. We've seen two examples.
Right.
Now there's a strong basis of evidence that this mutation is associated with cancer. So that database becomes better over time and becomes better over time of seeing results from lots of different people. So what has happened to this point has been that we haven't had as diverse a populace represented in those reference databases that when somebody goes in for genetic testing, they find a variant, then we go look in this database and say, oh, has this variant been seen before? That's how we're seeing that we get a lot of these unsatisfying results for Black women, for people who are not, say, of European ancestry, because they were not necessarily represented in the database. So they might have a mutation, but we don't yet know if it's associated with the disease or not. So not only is it unsatisfying, it's unsettling because, you're being told you have this mutation, but we don't really know
I know what it
causing
is.
your disease or if it's associated. And so one of the future contributions that Sambai would bring is perhaps bringing down that number of variants that are unknown or they might have been observed before, but we didn't know definitively that they were associated with breast cancer. And so we can say with the numbers that we're planning to profile in Sambai that we have enough evidence. There's thousands and thousands and thousands of women in our cohort, and we've seen this variant so many times that we're now more confident that this one is
There
associated.
is something there. Yeah, yeah.
So not only would it benefit our cohort, it will benefit future people who will get sequenced because now they'll have a bigger reference of non-European ancestry women to then compare to say, oh, these variants are associated strongly in this African genetic background with cancer risk.
Yeah,
Awesome.
somebody can help us answer a lot of questions or get closer to the answer. not even just for cancer, on a wider
Right.
base.
And
Yeah.
that's what being a scientist is all about. How can you really be a scientist and not have or want all the data? We had a scientist on our show once who talked about it was kind of a mic drop moment of not having all the all the data points. What is science if it's not a complete data set?
Right, if you have a piece of it. But I think that's what Sambai is. You guys help me out with this. I mean, I think that all of you guys, all of our scientists have been doing research kind of in our own little silo for many years or several years. And I think what Melissa has done here with Sambai is, okay, we're going to pull you from here and you from here and you from here and put us all in the same pot in the same, bucket to like, okay, let's all take what we've learned and then put it collectively together, and what can we learn collectively? I mean, don't you think, that's what kind of I'm saying from my little piece of the inside, that I feel like you're all such brainiac experts at your piece. So how do we pull all these pieces together and weave a quilt that's gonna give us better information about the disparities, right?
Yeah, I think she wanted to make sure she approached it from all angles, so people in all different fields of science, to give us a full picture
Yeah,
of a human and just really break down a human body.
But I would say, not just scientists. I think it's really critical and important in Sambai that we have you guys as well, and the voice of the community, voice of the patients represented and participating from the very beginning, to all the meetings. And as someone working in a genome center, as you can imagine, that's a fairly technical group of people. We don't even have patients. We are not an hospital.
right,
And so we talk about community engagements and trying to build that into our project, but it's the first time I really see it happening for real. And so that's really important, really new, and I think will really add a lot of value to the entire project.
great. All right, so here's a question for you. Just being a non-scientist, like, where do you get the blood from? Like, you keep talking about samples and samples and samples, and that's one thing as sort of our role. So where do where do you like, how many people are we going to get blood from? And like, where do we get the blood from? Like, help people
So
understand that.
the Sambai cohort is 40,000 patients. Okay.
So 40,000 people, which is huge, you guys. We're going to
Which
get
is
blood
really huge. Yes.
really huge, right?
I don't know if all of them are going to be cases, what we call cases, so patients of cancer. There might be some controls, and so some other patients that are not diagnosed with cancer, and that might provide some comparison possibilities. Usually, when people are recruited in this study, we discuss with them, we explain the goal of the study, and at that meeting or later on, there is an opportunity to get a blood sample, just a blood draw that can be done in the clinic. There is an alternative in some situations where we can get a saliva samples. That's what you explained before,
Okay.
and we can do the same analysis from saliva sample. That's fairly possible, straightforward, maybe not straightforward, but something that's very routine in most healthcare settings. The very much more difficult scenario is obtaining the tumor samples when we want to sequence the genome of the tumor. That's require a surgical procedure, a biopsy.
Biopsy,
The
okay.
storage of the sample is a lot more complicated. There's a lot of technical headache to solve to really obtain those samples, those tumor samples, and then characterizing them, sequencing them. But obtaining blood sample, it at least it's not supposed to be the difficult part. The difficult part is to have 40,000 patients in our cohort.
45,000.
That's
Yeah,
really a large scale, and that requires a tremendous effort from a lot of people in Sambai.
right. And those are samples are coming from all of our sites, which are the US, Canada, the UK, Ghana, Nigeria, South Africa, Ethiopia, and Kenya, right? So that's what we're
So
going
I think you have the
to
list, yes.
as of today.
as of today. So those are the areas that we're going to meet patients, and part of our job, Munoz and Laura, is to go make those patients feel comfortable and be able to explain what we're doing to them. So they say, can we like have some blood? We take a little blood test, or can we? And a lot of these patients I think we're going to encounter are already at the hospital because they're getting chemo, especially the cancer patients, right? So it's just a matter of, you're going to get your blood drawn anyway on Tuesday. So while you're there, do you mind if we take a little drip so we can analyze it? Right.
Yep, that's pretty much it.
I think another thing, Ricky, that's going to be completely revolutionary here is that not only is the cohort size very big, so 40,000, that's just a mind-boggling number, but there are going to be populations and whole countries of people who are going to have data on genomic sequencing at such a large scale that we haven't had before. And the data quality and processing will all be done in a uniform way. And
Okay.
so these data are going to be so incredibly useful in helping us understand, for the first time, for example, what is the relative contribution of your genetics, your genome, versus your experience in the world, your social environment, your exposure from the environment. And
Right.
not only in the US and not
Um,
only in the UK, throughout the whole world.
right.
All Black people, which has not been done before. All of this is unprecedented.
I think that's a really good thing to say. This has never been done before. What we are doing has never been done before. So that's how important this is. And I was speaking to a new patient this morning, and she has, ER positive breast cancer. I want to say it's the best kind you can get, right? If you're going to get this nasty disease. And she's stage 1. And she was asking me if she should get a double mastectomy. And I always say yes to that. Everybody who knows me on the show, like, do all the things, cut them off, do what you got to do, whatever. But I think that it's just so overwhelming, this disease, and it's so complicated to explain it, because she wanted to understand, as most women do, like, what did I do wrong? How did I get this? Did I not eat the right things? I'm not overweight. I didn't do this. I always eat vegetables, whatever. And so we're always trying to answer to them what the cause is, because they want to know for themselves, like, well, how did I screw my body up? Or why is my body failing me? And so I think what this is going to show us is we can help with that now. I mean, I think we're going to come up with some answers to understand, why did you get cancer? And we know that breast cancer is like one of the oldest cancers, right? I've seen pictures where they have like Egyptian doctors like holding a tumor, right? So we know we know it can't be prevented, but I think we're going to learn so much to help us be able to say, this is what happened. Or, so try to connect some of these dots that patients have when they're in this mess we're in, I don't know, but we have a lot to explain on what to do.
So, and one more thing I wanted to add on the discussion that that you might have with patients is, sure, yeah, we have to explain why we want to get a blood sample, but I think we also need to explain what we are going to do with the data, and
Yeah.
reassure them that the goal of this project is not to sell the data or to put it anywhere on internet and put it on Twitter and give access to that data to all the wrong people. And so we will be extremely responsible in analyzing the data, making the best we can out of the data, and sharing it with the right partners. So the goal is not to, get the data just for ourselves and not let anybody analyze the data, but really find the right partner to analyze the data, but keep it in very responsible hands. And so that's I think very important for the patients, that they need to be extremely reassured that we are not gonna do, crazy things with their data, and it's really very personal data for every patient. And
Right.
they really need to be reassured of what's going to happen to their data for how long and so on and so forth.
Thank you for
Right.
saying that. That's especially important in the Black community historically. We've been afraid of clinical trials and things like that because of how our data has been used in the past. We want to make sure it's in the right hands and it will be protected. So I appreciate.
That's awesome. Yeah, that's awesome, Dr. Nicholas. That's awesome.
How are you gonna come up with so let's say you get the blood, the saliva, and then you put it on this reader machine, the sequencer. How do you come up with the questions you want to ask the machine? I'm
So
just thinking about how we explain it to the community. And how does the community
Great question.
know
Great
the
question.
answer that you come, like the answers that you, the questions you ask and
yeah.
the answers that you get from this data that they've given their,
Mhm.
tissues to?
Go ahead,
So
Nick.
I would just start by saying that I think most of the questions we ask are of the cohort, of the entire data set, right? So it's not so much about the individual data, it's out of this whole, cohort of patients that are all diagnosed with a cancer. We can have information about their germline genome, have information about their tumor genome, have information about their exposure, have information about their occupation, their age, the number of kids they had, all of those information we have, and then start to ask questions. Okay, so if we have these type of profiles, does that correspond to that type of information in the genome, and so on and so forth. And so it's not so much like an individual question about one single uh, patients. It's about on a group of patients, do we see a trend? Do we see a pattern that, can give us more information about the way the cancer developed, for instance, or the reason why it developed, the cause in general of cancer. And then, we can track back to the individual patients and say, well, that happened to be the case in these patients, that patients, but not in that one, for instance.
Got it.
So that answer your question?
Mhm, mhm.
So you guys, before you started doing Sambai, can you give an example of something that you did where you had an outcome already? Like a success story, like something where you were researching it and this is what you found, just to give an example of like, how this works. Like maybe something that, and maybe something that gave you your purpose in this work. Like, well, how did you know you want to do this? Like, did you find something and like, oh my God, I found this and this is gonna do whatever.
Nyasha, you want to start?
Sure. So I don't have a smoking gun necessarily, but I have been in this field for a long time. Don't let the face fool you.
Oh,
But I think that I've been again, like we've touched on this a little bit. We've been studying cancer genomes for a while now, 10 plus or more years. And we constantly run up on this issue that we have people who are dying disproportionately who look a certain way. And then we have the people who we study, who look a different way. So it always used to bother me. Like, how come we're not
right,
studying the people who are dying from the disease the most?
got it.
So that was just something that as I went through my training and as I advanced in the scientific career, I was like, this doesn't really make sense to me. And then one of my
Love that. I love that. Say that again. How come we are not
how come
studying
we're
people
not
who are dying?
studying the people who are dying from the disease the most?
Wow, awesome.
It's really just that simple. That's what motivates me to get up out of bed every day. But the practicalities of it are much more complicated. As we can see in Sambai, it takes a lot for us to get this type of study off the ground, get all the people together and get samples and the
Right.
machine going. But I will always give a shout out to my postdoctoral advisor. So after you get your PhD, they send you to more school for like another few more years. For extra training. And so I was doing that and he just asked me, "What do you want to do?" And I told him, I was like, "This thing is bothering me and it's just I don't see how to do it in the field." And then he just said, "Well, why don't you just be the first? Just go do it. You be the one to do it." And so I sort of started gravitating towards changing what I wanted to do in my career was to make sure that in genomics, we were studying more diverse people so we could understand better how our genes are different and why we get sicker or don't do as well on certain medications. And
Mm.
that's what I focus on in my research. And if it wasn't for him saying, just because you don't see anyone doing it right now, you can't do it, just go do it. So there
Right.
wasn't any one particular study where we found the smoking gun yet. It will be Sambai. But it was just the pivot in direction and the motivation for it is kind of why I get out of bed every morning.
I love that. Thank you for that. I love that.
Nyasha, you might want to name your postdoc advisor because we both work with him and
Yeah.
you know, yeah,
Yeah.
he's a fantastic
My
scientist.
postdoctoral advisor's name was Ilya Shmulevich, and unfortunately, he did pass away earlier this year from
Mm.
amyloid, AML. But even right up until the end, he was still my biggest cheerleader. So yeah,
Well, he's still your cheerleader.
part of my why.
Yeah, he's still
Yeah, he's just doing it from
I
a different
think-
place.
monitoring your career, Nyasha.
I think you guys also keep him alive as you continue to do this work that motivated you and how he supported you through it. I think that's beautiful.
Yeah, it's awesome.
So I have a you know another way to answer and actually a smoking gun story. So early
Okay.
on at the New York Genome Center, we studied a very rare cancer of the liver affecting a young adult and adolescent. It's very rare, just a few hundred cases every year in the world. And by doing this whole genome approach, and I put C3, really not looking for a particular region, we one day identify a type of mutations in all of the samples that we had. So we had 10 patients, we had multiple samples per patient, and in all of the tumor cells, we identify a type of mutations that was not known before. And we, discovered and validate that finding in the same day. And we went home knowing that we had discovered something totally unique, totally new, that would
Wow.
completely change the trajectory for that disease. And
Wow.
we communicate with our collaborators. We wrote a paper. That was 11 years ago. And so for 11 years, I've been waiting for the time where that discovery will lead to a treatment. And so there are now clinical trials ongoing for this particular disease. But as
Wow.
Nyasha pointed out, there are still patients dying from that disease, even though we know exactly what type of mutations is present in their tumor, and very likely what is the early mutations that cause this tumor. So exactly how that works, exactly how to target that, exactly how to transform that into a treatment takes a long time, and we knew in advance that it would take a long time. But I can tell you I have alerts on all possible websites for the day where that would lead to a treatment, because I know that a treatment for that disease will be the consequence of that finding that we had early on. And so that's really a unique case of a eureka moment where really we can tell the exact day and time and place where we made that discovery, and having that incredible feeling of discovering something unique, and really sharing it quickly with our community, but really knowing something and going home and knowing that we have discovered something unique is something that justified the amount of work, the amount of studies that we've done to do this work, and that make the job of a scientist very unique. I can't imagine another job having that type of feeling. And I've been talking to high school students about that discovery just to give them the inspiration for pursuing this type of work.
That's awesome. That's awesome. That's awesome. What were you gonna say something? I just wish it wouldn't take so long.
Yeah.
Just think about, so you discovered this 11 years ago and we're just getting to the clinical trials of a solution, of a therapy. Wow.
Yeah.
Wow, wow, wow. How can we make it go faster, you guys?
I mean, in this particular case, the fact that it's a very rare disease is probably, a factor, right? If it was affecting a much larger number of patients, there would be a lot
Yeah.
more interest, right? So that's
Got
a
it, yeah.
completely, in fact, unfortunate economical situation that we're in.
Sometimes things need to be monitored over time, right?
Yeah. But you know, when I first got diagnosed with triple negative breast cancer, when I first started looking at the numbers, I remember I think Leah Scott did the first study, the first national study that she was at the University of Georgia. And I think she was still a student, working on her PhD, but it was the first time they said, okay, Black women get at 3 times the rate of white women. And that was like an aha moment for me, because I'm thinking, well, maybe we don't have any therapies for TNBC because it's a Black disease. I mean, is that what the problem is? Is it because only Black women get it more readily? And it made me think about, like, all right, money drives everything, including research, right? So where is the research money? Where is it going? And so that's why this is such a huge thing to get this money, this $25 million that Melissa got to support what we're doing. And it's it's an anomaly. It's not common. It's a blessing, right? I mean, you guys talk about how you get money and how, how does money fund what you do?
Yeah. I mean, I think another way to think about it is that actually things go fast, right? So we can, in the grand scheme of things, we discovered the double helix about 80 years ago, right? 1953, the publication of the double helix of the DNA. The genome that we talked about, those 3 billion letters, that was identified at the 2001, right? So 23 years ago. And so now we have the machine to read that, in a few days, to read a large amount of them, to analyze a lot of that. That's actually fairly recent, right? 15 years ago, we couldn't imagine doing the whole genome for 40,000 patients. The first cancer genome was less than 20 years ago, right? So that's, it's both very frustrating, in particular for patients that, there is no more treatment. But the rate at which we detect cancer, the rate at which we cure some of the cancer is actually accelerating. Um, you know,
Great.
2 generations ago, kids who with leukemia were all, dying in the hospital of Dana-Farber.
Right. Right.
And it's still very, very, it's still a terrible disease, but we can treat a lot more of them. Same thing for a lot of cases of breast cancer, as you alluded to. Some of the
Yeah.
subtype of breast cancer are much, much better treated than they were 20 years ago. So
Right.
there is a lot of progress. I think we always need to remember that there is progress. The progress is coming. And so it's always too slow, right? And we should all, make sure that it comes faster and there is more effort and money is better allocated, better used for more treatment. But it's not hopeless, and it has been going in the right direction.
There's a lot
Yeah.
happening behind the scenes that the community has no idea, and we're in the world of science, right?
Yeah, I think
That
we
happens.
should not stay in the world of science. That's the important part, right?
Yeah.
So that's where we need to translate that to
Yeah.
the patients. Absolutely.
So cool.
That's why we're here.
That's why we're here. So, gosh, a lot to learn today. Thank you for your giving us words we could spell. Thank you for what you're doing, and I'm so grateful to work with you guys and do this work. Was there anything else we should want to ask our lovely guests?
What? Maybe if anyone wants to learn more about genomics, any ideas, suggestions of where they could go for more information?
Some information on the New York Genome website. I'm sure, many other resources, depending on really what they want to learn. It's not, I don't know if our website is really targeted toward patients
Patients, right.
and cancer, and maybe we can find other resources that we can find we can add to the notes of this recording. And I think some of us in Sambai are always happy to answer questions and discuss more with interested people.
Yeah, you can always email. You guys know how to reach me, Ricky@touchbbca.org. And I'll, kind of pivot everything to my partners, and Laura's always, to Laura, what's your email?
Yeah. It's
Oh.
easy if you do hello@touchforlife.org.
Okay.
That's
Okay. Hello at Touch for Life. That's to Laura. And you guys, pretty soon we're trying to get it out, get it published by March. We're building a dictionary for patients, for people. And so we're trying to bring some of these complicated scientific concepts to words we can all spell and understand with examples. And so we're working on that fully digital, hopefully launch it by our Sambai meeting, by our Cancer Grand Challenge meeting in March. So that's what we're working on.
Happy to help in any way I can.
Yeah, we're gonna call you. Don't worry.
Good. Awesome.
We're using some AI, but we need some help. So we'll call you. But thank you so much for your time. I know it's very precious. We're very grateful for you doing this with us, but I think it's going to help the world know what we're doing. And we're so proud to be a part of it and do more of it. And Laura, I love you dearly. And she's our partner in crime, and you guys are awesome to work with. So thank you so much for joining us today for Sambai Speaks.
Thank you so much for having us.
Thank you very much.
All right, we'll see you next time for Sambai Speaks. Take care, everyone.